My name is Lillian Bischoff, and I'll be presenting a comparison of two fetal stem cells in proliferation, trilineage differentiation, and influence of hypoxia. First, let's discuss some key terms from the background. Tissue engineering is a field that uses stem cells and other biomaterials to generate functional tissue. Stem cells are cells with the ability to develop into many different types of cells through cell differentiation, which is the process by which a stem cell develops into a specific cell type. We will be discussing three different cell types: adipogenic differentiation, which results in fat cells; chondrogenic differentiation, which results in cartilage cells; and finally, osteogenic differentiation, which results in bone cells. Lastly, proliferation is cell reproduction. As a brief introduction to this project, articular cartilage possesses limited ability to self-repair due to a lack of blood supply. The current procedure to repair cartilage damage is autologous cartilage implantation, which is re-implantation of cartilage from within the body. However, this has several limitations, so the field of tissue engineering has emerged as a possible solution. Two cells that are viable candidates for cartilage tissue engineering are SDSCs, synovium-derived stem cells, and NPCs, nuclear propulsive cells. In our study, we hypothesized that fetal SDSCs and fetal NPCs would possess distinct proliferation and differentiation potential for each of the three lineages: adipogenic induction, osteogenic induction, and chondrogenic induction. We also hypothesized that a low oxygen or hypoxic environment would improve the chondrogenic potential of fetal stem cells. A brief overview of our methods begins with the culturing or growing of the SDSCs and the NPCs via a standard protocol on plastic flasks. Both types of cells were induced for adipogenic, osteogenic, and chondrogenic differentiation with appropriate mediums, which is the food for the cells. Then, the chondrogenic differentiated groups were each split into a hypoxic environment with 5% oxygen and a normoxic environment with 21% oxygen, so that we could assess hypoxia's effect on the cell's potential to become cartilage tissue. Finally, analysis methods were used. Flow cytometry was used to determine proliferation capacity and to identify surface marker expression. Oil red O staining and alizarin red S staining were used to determine different adipogenic and osteogenic differentiation capacity, respectively. Real-time PCR will be used to quantify differentiation potential for chondrogenic marker genes. Looking at the results in figures 1 and table 1, the NPCs were shown to proliferate about 1.5 times the median rate of the SDSCs, meaning that they grew about 1.5 times faster. Secondly, the surface marker analysis results are shown in figure 2. To briefly summarize these results, the surface markers were shown to be very similar between the NPCs and the SDSCs. Both cell types stained either positively or negatively for markers in correlation with one another without significant differences. Figures 3 and 4 display alizarin red S staining results, where the red stain displays calcium in the matrix surrounding the cells after 21 days of osteogenic induction. Figures 5 and 6 display oil red O staining of the NPCs and the SDSCs, where the red stain displays lipid droplets within the cells after 21 days of adipogenic induction. We can draw some conclusions from all of these results. First, NPCs proliferate more rapidly and share similar surface markers to SDSCs. Second, oil red O staining demonstrates SDSCs have a greater potential for adipogenic differentiation than NPCs. Third, alizarin red S staining demonstrates that NPCs have greater osteogenic differentiation potential than SDSCs. Finally, preliminary evidence suggests that hypoxia might improve the chondrogenic differentiation for both stem cell types, as the pellets grown in hypoxia were actually qualitatively larger than the pellets grown in normoxia. As the study is in progress, we expect that the PCR data will exhibit distinct chondrogenic differentiation potential for each cell type. Considering broader impacts of this research, the continued study of cartilage tissue engineering may provide a novel approach to regenerative medicine, particularly for patients with articular cartilage injuries. Additionally, the determination of the optimal cell type and environment for cartilage tissue engineering will play a major role in the future success of cartilage repair procedures. For future work, the extracellular matrix components, as well as surrounding environment, should be further studied for the impact on each stem cell's ability to become cartilage tissue. Lastly, in vivo studies are necessary to determine the impact of implanting tissue-engineered cartilage grafts into the human body. Thank you for your time.